All right. Good afternoon, everyone. Welcome to the weekly Living Earth Collaborative in Washington University EEPB seminar series. Today, it's my pleasure to introduce our speaker, Dr. Matt Dugas, who is currently an assistant professor in the School of Biological Sciences at Illinois State University. Dr. Dugas graduated from University of Oklahoma with a PhD in zoology. He then worked at Hendrix College as a visiting assistant professor for a year and did a postdoc at Tulane University, where Dugas and I briefly overlapped. I was a first-year PhD student, and he was the person that showed me around the poison frog field sites down in Panama. That was during my first field season. And after Tulane, Dr. Dugas worked as a research associate at Case Western Reserve University, where he overlapped with Mike Moore, and then a postdoc at Murray State University before moving to his current position as an assistant professor at Illinois State University. Dr. Dugas's research focused on the evolution of animal communication and life history trade-offs. He has worked on various vertebrate systems, including birds, frogs, and fishes, and one of the focuses of his research is on parent-offspring conflicts and communication, which I believe he will tell us about in today's seminar. And so before handing the mic to Dr. Dugas, I just want to remind everyone again to leave your comments and questions in the YouTube chat box anytime during the talk, and we will relay the questions to Dr. Dugas at the end of the seminar. And so Dugas, without taking up more of your time, please take it away.
All right, thanks, Yusan. Appreciate the intro. Hi, everybody out there. Thank you for having me. Share my screen here. This is a special treat for me because Yusan and Mike are hosting. So this is really a delight. As Yusan said, I met both of them in their first years of PhD programs, and now they've both greatly surpassed me in scientific excellence. I'm glad they'll need my mentorship again in the next year or so for student emails and faculty meeting. So I stand ready. Okay, so let me get going. I'm going to talk to you, as Yusan indicated, about begging in two different systems. So before I get really going, I want us to sort of get on the same page with what is going on in a real family in nature. So when we think about families, we often have a picture in our head, something like this, right? Where there's plenty of food, everyone seems to be getting along pretty well. But in nature, for various reasons, families look a little bit more like this. So there are lots of evolutionary incentives for parents to initially produce more offspring than they intend to rear. And then even among those offspring they're intending to rear, there are some conflicts of interest that select for offspring to want a little more from their parents than their parents really want to give them. So within real families in nature, resources are limiting for environmental reasons and for social reasons. So what does a parent do in the face of resource limitation? So this parent's got one food item and it's got 4 hungry mouths. So an easy way to think about this is with Hamilton's rule, where we think about the parent delivering that food item, and it's going to be selected based on its coefficient of relatedness with those offspring, the cost that it's paying for that food item, and the benefit that each offspring is going to get. And if you don't like thinking about Hamilton's rule for parental investment, just ignore the R, and it's an issue of where the benefits are going to exceed the costs most. So from the parent's perspective, all else being equal with respect to relatedness and that cost, the thing that's different about those offspring is that B term. So when this parent looks at this, it's already paid the cost of acquiring that food item. At this point, it doesn't matter anymore. So the only way an offspring can beat, or a parent can beat, just delivering food randomly is to attend some way to the benefits that each offspring is going to receive from that prey item. That's the only way a parent can do anything better than just randomly delivering food. So a broad hypothesis is that this B term, this benefits that the offspring is going to get from that food item, is what's driving the evolution of traits that we usually call begging. So you're probably familiar with this in birds, where the nestling bird's going to open its mouth, it's going to stretch, it's going to wave around, it might flap its wings, it's going to make some noise. This is also common in mammals, where it looks a little different. It's more of a noise and a less stereotyped behavior, but it's recognizable. There are invertebrates that do this. Burying beetles, certainly the best studied, where the larvae will sort of rear up and wave in the air. Bee and ant larvae do this too. Earwigs, nymphs will produce a chemical signal. There's even analogs in plants where some chemical communication between the developing embryo and the parent plant is mediating the exchange of resources. Of course, young humans do something like this as well. So there are basically, when I got working in this, there were two general hypotheses for what this means. All we know is that it has something to do with that B term, those benefits that offspring are going to get, but what kind of benefits they're getting and how that shapes the evolution of signaling, less clear. So if you know something about begging, you probably know the signal need hypothesis. So this is the hypothesis that offspring in relatively bad condition stand to gain a lot from that food item. So if they're not doing well, the fitness increase associated with acquiring a food item is really high. So parents get a lot of marginal return by directing resources there. So we see signals evolve to be negatively associated with current fitness prospects. That's because, again, they stand to gain a lot from getting that food item. An alternative to this is probably more familiar from other signaling contexts. And this is the signal quality hypothesis. This is basically that signals in a young dependent offspring are selected for the same reason we see signals all sorts of places, like male signaling to females, right? And that's that this is indicating, hey, I'm doing really well. I'm a good place to put further investment. So if this is right, we expect signal intensity to be positively associated with an individual offspring's current fitness prospects. So up until probably about a decade ago, almost everything you saw was about the signal of need hypothesis. And the signal quality was sitting out there. There are some results that sort of support that, but not really formalized at that point. So studying signal equality, one of the things we want to do starting out is just find a good candidate trait, right? So this idea makes some sense. So what's a good candidate trait to look for to function this way within a family? So I'm going to be telling you about candidate trait of colorful nestling mouthparts. So nestlings in lots of birds display yellow, orange. We'll talk a little bit about the diversity, but they display these colorful mouthparts. And importantly, in a lot of birds, this is only during the nestling period. So a robin's not a good example because adult robins also have these yellow mouthparts. But here's a picture of a cave swallow. So we have a male over here in that top left panel, and then a female, a little bit different. But they're both very different from nestlings. So this is just a range of nestling coloration you see in this species. And nestling mouths look very different from adult mouths. One of the things we're doing now in my lab is I inherited a truly impressive collection of frozen birds. And so we're going to try to go through and quantify what mouth color looks like in adults so we can figure out how much of this is specialized, how many species this is a specialized nestling trait. But in a lot, it is. And so it's a little cheesy, but I want to kind of give credit here to research that really writing that happened about 100 years ago. So this is where the study of nestling mouth coloration really got started. So here's a plate from that, and you'll see the quote, but basically the argument here is that, hey, there's lots and lots of variation here. There's lots of variation in color, there's lots of variation in patterning, and these are colorful traits that are comparable to other colorful traits that we see in nature, and we should pay more attention to this. And I'm going to argue that despite a lot of work on this, this still is true 100 years later, that it doesn't get the attention it deserves. Okay, now, despite this argument that I just made about how there's all this diversity, I'm now going to say, well, yes, but we can really collapse most of that diversity into about 3 parameters. So what you see over here on this panel is you see a photograph and then a spectral reflectance curve of the flange tissue, which is this part that outlines the mouth of a bird. And then that's always in every single one of these, this is that higher curve. And then this lower dark curve is the reflectance of this interior gape tissue. So these colors are different. There is variation, but the variation is pretty constrained. So this is a reflectance curve from a house sparrow. So again, up here is the flange tissue. Down here is the gape tissue. Not going to talk much about the gape today. In house sparrows, it's not that interesting. You can see little dips in reflectance that's associated with hemoglobin. This is mostly a blood-based tissue here. So this flange tissue is going to be better reflective of sort of what variation you see across and then within species. So they differ in how yellow they are. You can see that on the pictures on the side panel here. So what that yellow comes from is carotenoid tissue or carotenoid pigments in the tissue. This are, as far as we can tell, there's not a lot of variation in what carotenoids it is across birds. It's the same things, and it varies in concentration. Carotenoids color things by subtracting light, so they absorb these middle wavelengths, these greens and blues. And so when carotenoids are in the tissue, you get this dip in the 400 to 500 nanometer range. What that does is the steeper that dip, the deeper that valley, the more saturated, the more intense that color is going to look. Colors also differ in their brightness. So in the brightness is just the total area under that curve. So how much total light is being reflected from that tissue. This again, there's some variation within species. If you look over on that side panel, you'll see there's quite a bit of variation among species too. And then finally, they differ in the extent to which they're UV rich. So lots of different ways to quantify it. They're all correlated with each other. The way I quantify it is by comparing the reflectance over in the UV range, so 300 nanometers to 400, well, 3 to 320. And then over here on these long wavelengths, that should be just kind of an indicator of the maximum brightness of the tissue. So the question there is, how bright is the UV part compared to how bright it should be if it's just flat reflectance? So these are, again, the three ways that tissues, the mouth parts of nestling birds, seem to vary both within and among species. Okay, so I want to talk to you about what this variation means in the study organism where I spent most of my time, the house sparrow. I don't know, I can't see the list of attendees. I don't know how many people are shaking their fists at me now for house sparrows, but they're a great study organism for this topic. So they're a globally distributed bird at this point. They're non-native to the United States, but they're everywhere. They form pairs during the breeding season. There's some extra pair mating, some males are polygamous, but generally it's a social pair. They readily use nest boxes. They don't like to be around trees or dense shrubbery or things. So usually you have a parent flying into a nest, so a nest box in this case, a dark nest from being out in the open. When they come in, they're faced with nestlings that look something like this. So they have this really clearly defined yellow rictoflange, and again, that blood-based gape in the interior of the mouth. Okay, so what does variation look like in this bird? So over here, you see a really pale looking nestling. This is what nestlings look like when they hatch. Pretty much everybody looks like that when they come out of the egg, and they look like that for about a day. On the other side here, you see a lot of yellow coloration. This is a nestling that's only 3 days old. This is an outlier. This has put on quite a bit more color than your average bird, but they all get yellower as they get old. So what we see is between days 3 and 6, and this is of a 14-day nestling period. So this is about as young as we can really use a spectrometer to measure color. And then by day 6, they're starting to sort of jostle around and compete for favored sites. So this is the range where parents really have a lot of control over resource allocation. So during this period, we see color is changing in a reliable way. So those tissues get less bright, they get less UV intense, and they get yellower. So they're packing on carotenoids during this time. So there's some change in the tissue, and what happens in that tissue is that they get yellower, and that causes them probably to get less bright and less UV rich. Okay, so what about on a day where parents are going to be interacting with their offspring, right? So what a parent's going to see. And this picture I'm going to leave in the corner the whole time. That's not out of the normal range of what a parent might see on one day when they come to the nest and they have a food item and they're going to deliver them to their offspring. So if we look at some measure of individual quality and color, do we see any sort of relationship? If this is going to be a signal of quality, we need to see some sort of relationship between the signal intensity and some quality metric of the individual. So here's our three color metrics. Chroma is the yellowness, brightness, and the relative UV intensity. And what we see is if we just look at this tissue, this right here is mass and total carotenoids. So a bird that's very yellow is heavier and it's circulating more total antioxidants. So this is just a simplified version, but we've done this with parasites, hematocrit, a few other metrics of fitness. And what we basically see is the more carotenoids there are in that flange tissue, the better quality that individual bird is. So I promise this will become sort of relevant at the end of this, but I want you to notice that brightness, at least if we look at it now, there's nothing there. And this relative UV intensity, if anything, it's negatively associated with the whatever quality metric we can use. Okay, remember I told you that carotenoids are subtracting light, and that's how they're generating color. So it's kind of expected that by subtracting light, they might influence that total brightness. So when we statistically address that by just including that yellowness in the model, we see that all of a sudden brightness is positively associated with these quality metrics. And again, this is true for other things too. It's just, it's the same story no matter what I show you. And then for UV, there's nothing there. Right? So the underlying brightness of that tissue is suggesting a good bird. Although again, if the parent, as the parent sees it, there's nothing there. Okay, just one more piece of evidence that these two things are doing something important. They're containing some sort of, they contain information. We don't know if they do anything yet, but they contain some information. Something called the nutritional hypothesis is just this idea that signals and their underlying quality become associated because nutrition is driving both of them. And so to get a look at this, we just look at the color change over that day 3 to day 6. And we ask how much is that color change associated with how much mass they gained. So did birds that gain a lot of mass over that time also change color a lot across that time? And indeed, we see that that's true for, again, that yellowness, that carotenoid richness, and for the brightness of that tissue, the underlying brightness of that tissue. But there doesn't seem to be anything going on with UV. So another piece of evidence indicating that chroma or yellowness and brightness are reflecting some sort of quality metric, and they're integrating some sort of information about how well this bird is doing. Okay, so just checking in on where we are in this first part. If this is going to be a signal of quality, we want to see some positive association between fitness prospects, some measure of that, and signal intensity. And indeed, we see that. We see that for at least 2 components of this color. So what about whether parents respond to this? They can have all the information at once, but if parents don't care, it's not evolving in the context of communication between parents and offspring. Okay, so rather than just asking what parents respond, whether parents respond, I kind of want to break this question down a little bit and say sort of why approximately parents might respond. And I'll just say signals generally are going to contain 2 types of information. The first thing they do is they tell you there is a sender around, and the sender is somewhere in some limited range of physical space. After that information is conveyed, you can also have more quantitative information, so about sender quality. And I think this first part is the less sexy answer, and so this doesn't get a lot of attention. But this is a really important part of understanding hypotheses for why nestling mouth colors evolve. So the detectability hypothesis, which is nothing but the name I gave it, is really put most elegantly, again, over 100 years ago. And this is a hypothesis built on the assumption that putting food in a nestling's mouth is no easy matter. That a parent bird has a really difficult task to do. It has a big food item, a small nestling. This nestling's not great at swallowing stuff. It's got to put the food item right in the right place. It's often got to do that quickly with a lot of things moving around. And it's a lot of birds, including house sparrows, have to do this after a transition from light conditions to dark conditions. So this was the hypothesis to explain this phenomenon of nestling mouth coloration when it was first discussed. And it's really a simple one. It's that this nestling mouth is nothing but a target. So it's not a signal of quality. It's nothing fancy like that. It's just a target for putting the food in and getting the food in the nestling's mouth. Okay, so how do we know if colors are easy to see for a bird? One approach you can take is visual modeling. And so this was the first thing that I did on this to try to get a hold of this question. And we just looked, are these more yellow nestlings, the nestlings that appear to be in better condition, are these easier or harder for parents to see? You can model what this would look like in realistic lighting conditions with the visual system of the parent. And you just ask whether these things are related, how they're related to the intensity of that coloration. So it's a little blurrier than I imagined, but the take home here is that we did this in all sorts of lighting conditions, all sorts of directions, all sorts of times a day. And what's really going on is there's these two ways that colors can contrast. They can do this in the achromatic channel, which is really just brightness differences. And those are probably what's important for detecting things at low light. And in here, you have basically minimal relationship. If there's a relationship, it's negative. With respect to color contrast, the yellower that flange gets, the less different it looks from the red of the gape. So if there's any effect, it's negative, it's a weak effect, there's not much. And if that doesn't make any sense to you, just look at this. This is just a dramatization of what I'm talking about, where you see a pale nestling on top and a yellow nestling on the bottom. And we just look and see what happens as we make that picture darker and darker. And we see is it's a little bit easier to see that pale one. But I'm unconvinced that this is the real solid answer and we can't do any better. So I came up with a behavioral assay to try to come up with some way to quantify how hard it is for the bird to really see and do what it needs to do. So here are nestlings painted to maximize and minimize achromatic contrast. So this one's got white paint on its flanges, and this one's got gray paint that matches the brightness of the tissue inside the gape and the skin of the bird. They're also both fitted with like a collar, like you would see on your dog after it goes to the vet. So they have this little collar, so when they're begging, the flange is presented against black felt, so that we really can maximize the differences between these two treatments. We can make one really easy to see, one hard to see. So parents interact with size matched nestlings, one painted to be gray, one painted to be white. Okay, so I'll show you what kind of things we can quantify. This is an easy parent visit. So the parent shows up, drops that prey item off, and we're done. This parent over here is going to have a little more trouble. So it's going to come in, try once, twice, 3 times, 4 times, 5 times, 6, and 7. And now it's delivered that prey item. So we have an easy visit, one attempt, very quick, and a visit that took a long time and needed a lot of attempts for the parent to get that food to actually go down. Okay, so when we did this, what happened is that we had found that those ones that should have been harder to see were harder to feed. So it took longer to feed, for the same parent to feed a nestling painted to be very inconspicuous, than it did for the same parent to feed one that was painted to be very conspicuous. This is true for total duration of the event and the number of attempts that it took to transfer that prey item, measuring the same thing. Okay, so now we have behavioral assay, proof of concept here, something matters, but within that natural range of variation, what do we see? And so we're going to ask two questions about that natural variation. One, how easy it is for parents to see that mouth, so use that behavioral assay, and then whether parents care. Is there evidence that if this contains information, that parents like the information or like the signal associated with that information? Do they like really good quality colors? Do they like not so good quality colors? Do they care? So this is work that really got picked up by my PhD student, Shayna Porter, and she's had help from two great undergrads in the lab for a couple summers now, getting through all these videos. So Shayna did a great job, was very meticulous, the level of patience I would not have had to really match natural colors. So you're seeing here pictures of a model. So this is our really yellow flanges in the in a population, so the yellowest in our distribution, and then a paint. You'll notice the UV is not perfect, but that's really, really hard to do. And then for the pale, the same thing. The pale's a really good match. So if we look at our objective color metrics, they're almost identical except for UV. But the good news is that there's not very much UV in that box anyway. So when we do that visual modeling, the contrasts between the colors and the things it might be posed against are basically identical to what they are for the model colors that we use. So the paints look just like these extremes of flange coloration would look. Okay, just to again give Shayna a lot of credit, I did this a decade ago, and you can see natural colors and the paints I used. So that UV is not easy to do because a lot of pigments absorb UV. So what we ended up doing is adding in Teflon powder that's a bike chain lubricant. And that managed to get the reflectance in that UV up enough that it didn't look weird to the birds. Okay. The second part of what we did is we made paints that would hold yellow constant, and then we just manipulate those other aspects of color, brightness and UV. So our brightness paints, they look like the same yellowness to the bird, but one is much brighter than the other. And then to block the UV, we essentially put on sunscreen. And this is a really standard thing. It's not maybe as nice as it could be, but it's what everybody does when they block UV colors. And these colors, again, when we look at them, they look good. They look just like the colors, except they're different in this one parameter. Okay, so when we let parents choose between something that looked really pale and something that looked really yellow, they gave more food to the one that looks really yellow. So this is really the important check on that signal quality hypothesis. So this is the results from last summer in Illinois. So we see that parents are allocating more food. We restrict these analyses to cases where both nestlings were begging. So the parents actually choosing between those two colors. And this matches what I got even with my subpar paints a while ago in an Oklahoma population. So this was a great relief to me that this paper that I had been resting on for a while, this actually held in another population. Okay, but it doesn't seem, even though the visual modeling suggests that this yellow coloration is maybe negative for visual conspicuousness, it made no difference to the birds. So these are just two different prey sizes. This is the original paper with prey size called them tiny, medium, and enormous. So I use that now. So really small prey items and even these medium-sized prey items made no difference. So the parent could deliver that prey item just as easily to a really yellow chick as a really pale chick. So they like the yellow, but it makes no difference to how easy it is for the parents to see. Again, it may be mathematically easier to see the pale one, but it makes no difference to the parents performing the actual task they need to perform. All right. Brightness, UV richness, parents do not care. Right, so it makes no difference to a parent whether it's interacting with a bright nestling or a not so bright nestling with respect to food allocation. So here again, you see these dark bars are instances where both nestlings were begging. It's basically split down the middle. So some parents fed more to the one that was brighter, some parents fed more to the one that wasn't. It didn't seem to matter. Same thing is true of UV. So it didn't make any difference to parents. They allocated food equally with respect to those two color treatments. For UV richness, it doesn't seem to make any difference for detectability. So here we had, just because this manipulation lasts longer, we had more of these really large prey items. We had enough to include. But you can see it takes parents longer to deliver large prey items, which I found before too. But there's no effect of this UV treatment on how quickly they transfer prey items. Brightness is a little trickier story. So with respect to prey transfer efficiency and brightness, what we see is that for ones that were not so bright, the dull treatment, it mattered how big the prey item was. So it took longer to deliver that bigger prey item for that dull treatment, but not for the ones that had that especially bright flange treatment. So brightness seems to matter, but conditionally on the size of the prey item. And I'm going to apologize for a boring slide, but years ago, I found this three-way interaction, and I've never had the courage to go back and deal with the analysis formally. But what I did was I measured prey transfer efficiency, how long it took parents to transfer prey during the day when natural lighting conditions vary. And there's a three-way interaction between prey size, nestling size, and ambient light. So the punchline here is that ambient light matters for prey transfer when food is large and nestlings are small. Other than that, it doesn't really matter. So broadly, this is another piece of evidence on that pile that parents are sort of visually constrained sometimes in transferring prey items. Not all the time, but there are some conditions under which they're visually strained in the natural lighting conditions. So I'm just going to take a second to pause and sum up what we have here with house sparrows. And I hope convince you that even if I've bored you up to this point, that this is going to be something worth thinking about more broadly. All right, so these nestling house sparrows are variable with respect to their yellowness, their brightness, and the UV richness of that tissue. Okay, so first let's get rid of this UV thing. This UV doesn't seem to contain any information that's not already encoded in carotenoid richness, and there's nothing that it does, obviously, right? There's nothing we can see that it obviously does in, at least in house sparrow nests. So what I think the explanation is here is that some of this is just people got caught up in the discovery of UV light and that birds could see UV light. We all got a spectrometer and we measured it, and that was great. But probably what's going on here is that there's selection on nestling birds to just be bright, right? To just have this broad reflector. Birds can see the UV, so it includes, their white includes UV, no surprise there. And you get what looks like a UV peak just because you have carotenoids sucking out those middle wavelengths. So it could be that selection for a broadband reflector. It could also just be, hey, this is what the tissue does. You know, Teflon tape reflects UV light, not because that is important to its function, it just does. But including some of my own stuff in swallows, like it doesn't look like this is true for every bird. Some birds seem to care when they're allocating food, and it does seem to contain some information in other birds. So I think one direction to go from this finding is to sort of start asking approximate questions about how this does get co-opted in other birds. So what's different about the UV reflectance, how that's generated, how that's sensitive to development? What's different in house sparrows and those other birds where it does seem to contain some information? Okay, so larger, of course, the key take home, the simple one, is that larger, better, whatever nestlings seem to have yellower flanges in house sparrows. And this is pretty easy to explain because parents like it. So parents like it, so offspring display it. Whatever one of those came first, doesn't really matter. That's our explanation for how it functions at this point. And it makes sense that it would be maintained. It makes sense that it could have evolved along with a parental preference and parents benefiting from feeding those high quality offspring. So any costs of signal production paid to make a mouth yellow are returned to the nestling in the form of increased parental food allocation. A little more complicated for this brightness one, right? So one of the approximate things that seems to be going on is that the bigger nestlings don't actually have flanges that appear any brighter, but they should have flanges that appear darker because carotenoids are sucking out light. The reason they don't, and the reason there's not a strong relationship between carotenoid richness and visual conspicuousness, is because those high condition nestlings are also doing something to the underlying tissue that compensates for that loss of brightness. So carotenoids are sucking out light, they're somehow making their tissue brighter, and that equals out. So it ends up looking no different. This one's a little trickier to explain because the costs of this production are going to be borne by the nestling, and it can't be free or everybody would read really bright. And so the only way that they recoup that is by increased efficiency of parental prey transfer. So we have one component of the signal, the yellowness, that makes total sense, and they're competing for parental feedings. We have another one that doesn't make quite as much sense right now, or is a little more tricky to explain anyway, because the only thing this offspring gets out of that being bright is indirect benefit. So its parent can deliver food more efficiently, which pays off in terms of siblings getting more food, current and future siblings getting more food. So we could have an example in here, which again, I hope I convinced you. I know talking about the different color parameters can be a little numbing, maybe, I don't know. But I think this is really important, and I'm really glad that Shana went and did this follow-up work, because I think it tells us that we have two different parts of that signal that are maybe being selected for in different ways. Okay, so just sum up this part of the story, and I'll do my second part probably pretty quickly. We were looking for something that was a good candidate to work through this hypothetical signal quality, and we got it. So there's part of this color that's positively associated with the nestling's current quality, and parents like that. The fact that parents like that should be driving the evolution of more elaborate signals. All right, I'm just going to take a second to pause for myself, and then I'm going to tell you a little more briefly about a second system where I've been working in, and that's poison frogs. So this is strawberry poison frog. You may have already heard about this from USON at some point. So what they do is they'll court, and then they'll lay eggs in leaf litter where the male does something, maybe takes care of them. And then when these hatch, mom picks up these tadpoles and takes them to small bodies of water. This can be the water held in a bromeliad, most classically. The females are going to come back every couple of days for about a month and a half and feed that developing tadpole with unfertilized trophic eggs. So mom's going to come back by herself. The tadpole is going to be there. She's going to feed it. And what's really important for us is that typically it's just one tadpole in each nursery. So a mom may have a brood of multiple tadpoles, but they are physically separated from one another. And what that means is that a parent that wants to be choosy in this scenario has to get its information from things that offspring are revealing. This could be via a cue like body size, or could be through some sort of signal like begging. One of the critiques of the signal quality hypothesis has been that, hey, if a parent wants to give food to its best offspring, it doesn't need to do anything. You can just throw some food in there and let them fight it out. Or in a sparrow, it can just sort of feed from the hole into that cavity nest, and then just let the chicks fight out for who gets there, or just feed it to the one that's closest to that hole. That'll get you the biggest one. You don't need a signal. So that's been one of the big critiques. This is a system where whether that critique is totally valid or not doesn't matter, because that is not possible here. Right? So parents, not only they cannot just throw some eggs in there and let everybody fight it out, And they also can't even compare their offspring at the same time, which would be something called a signaling scramble, where they could just pick the highest value when looking at everybody together. So when moms visit, tadpoles do something that looks like begging. This is going to be a little hard to see. It's dark in the jungle, but we see a tadpole here, and it's going to kind of swim around. At some point, it'll stop here by the mom, and then it'll start just vibrating in space. It's not really moving anywhere. It's just vibrating. So let me show you over here. This is slowed down. So this is a quarter speed. What this tadpole looks like in a lab assay of begging. So it's just sitting there, and then it's moving its body back and forth really fast. Remember, this is slow motion. This is not full speed video. You can see it sort of shaking its body on that axis. Okay, so a bunch of life history work that Mike sat through me writing up. We show that care is valuable to tadpoles. It's good for tadpoles to eat eggs. It's all they eat. So kind of no duh on that one. This care seems to be costly to parents in terms of future reproductive events. And food's limited within a family. So I'm going to tell you really quickly about an experimental manipulation of need, quality, and this third component, hunger. And just to review what we'd sort of expect here is if the signal of need hypothesis is operating here, we're going to see our old big tadpoles begging less. We're going to see experimentally high condition ones begging less. Maybe we're going to see our food deprived ones begging more, maybe not. Quality, opposite predictions are true. And then it could be that it's just about hunger. So to do this, brought in field collected tadpoles and fed them on a realistic regime, so something that might match what parents would do. They got 2 eggs either every other day or every 4 days. After they had done this for a while, when they were both ready for a meal, we looked at them again, and this was a really nice condition metric. They weren't more developed, they were just bigger for their developmental stage. Then we fed everybody as much as they would eat. This was really important because we were able to separate hunger from condition. So our low condition tadpoles ate more at this point. We assayed their begging when they were full, food deprived them for a few days, assayed begging again. So we got them when they were full and they were hungry. And then after this, we seem to have gotten rid of any residual hunger effects. There was no difference in how much they ate after that. So what did we find? First, very surprising if you work on birds or anything else, hungry tadpoles don't beg more or more intensely. So if, in fact, hungry tadpoles beg less and they vibrate more slowly. Old and high condition tadpoles beg more intensely. So they're able to hold their begging longer before they have to kind of take a break. And this is true of our high and low condition too. We have sort of complex effects of condition and hunger, where basically what's going on is our high condition tadpoles don't get tired as fast as our low condition tadpoles. So everybody gets tired when they're begging, but not as much if you're in good body condition. So all these signal production things make it look pretty clearly like begging is a signal equality in the system. What we don't know, and we're still really working on, is how much parents care. So we know in captivity and in the wild, older tadpoles get bigger meals for mom, which is certainly consistent with the observation that bigger tadpoles are begging more. Bigger and older tadpoles are producing more intense begging signals. With observations of mom in the wild, we see that even controlling for that difference in size, tadpoles that beg more intensely, that vibrate faster, get fed bigger meals. So it looks pretty good, but certainly not going to tell you that it's conclusive at this point. Manipulating begging, really hard. So one way you can do it is swap out a mom's tadpole for a tadpole of a completely unrelated species that does not beg. And when you do that, moms don't feed the tadpoles. So this tadpole's not begging, mom just kind of sits there. They sometimes go in and out of the cup. They don't know what's going on. They rarely feed them if they don't beg. Can we explain this? And I'm going to say, yeah, we can explain this really well. So in an experiment Mike was involved in, We removed tadpoles from their moms at various stages of development, and we followed them through 90 days post-metamorphosis, which is where they're about adult size. They may not be reproducing, but they're about adult size. Think about this as your classic relationship that's hypothesized between parental investment and offspring fitness. And the reason parents care about where their offspring are on this curve is that you see these decelerating benefits. So we can look for this by comparing the probability of surviving to 90 days of tadpoles that were taken away from their moms at some developmental stage and one developmental stage later. And that gives us the value of care during that stage. We know that older tadpoles get more, so we can just go in and adjust for that and get the fitness value of each egg. And when we do this, we saw a relationship that didn't look like this hypothesized one. We saw that those benefits are actually increasing. So maybe this is the real hypothetical relationship, but what actually occurs, this is in captivity. So even in captivity is just down here. They never get to that asymptote. So in this system, a signal equality makes perfect sense because parents clearly benefit from allocating resources to high quality offspring. And I think more generally, it tells us that maybe parents are sort of working close to the minimum, not really the maximum. Spoiler alert, this kind of works well for sparrows too. So here's the relationship between the mean mass of a brood members for a parent house sparrow and the predicted recruits that it's going to get from this brood. And we see the same relationship. The bigger those are, the more likely the parents are to get recruits out of that brood. And so in house sparrows, brood reduction is really common. 40% of the broods lose somebody, and this happens well before peak demand. This happens about day 4. And this is true in Illinois too. It's basically the same pattern. And so in terms of parental fitness, this makes total sense. As you can see, just making your brood 2 grams bigger totally makes up in terms of fitness of losing somebody. So we went back and tried to do this with younger house sparrow nestlings. So before that big crunch of brood reduction. So in those house sparrow nests, heavier nestlings and yellower nestlings gain more mass from days 1 to 3. And remember, 4 is when that mortality occurs, or before 4. This is also true for mortality. So heavier and more yellow nestlings are less likely to die across that range. What we don't know is sort of which is more important because those things are really highly correlated. And so the next step in this is going to be to let parents choose when color and size don't really match. So let me wrap up here and just say the big take home is signals equality seem to have evolved in 2 very distantly related lineages. So can signals equality evolve? Yeah, they obviously, because they have, and it's not some fluke, it's evolved in 2 really different lineages. Broadly, I think the answer to sort of understanding how signals evolve in a family is understanding those fitness returns that parents get. And maybe that's gotten lost a little bit. Sort of more nuanced steps that we can take forward, I think, is in birds. We can think about how the ambient light environment is constraining the evolution of these ornaments. So broad pattern we see is that open nesters have more intense colors, more carotenoid-rich colors than cavity nesters, with dim nesters intermediate. And so it's not that a bluebird wouldn't benefit from knowing more information in that mouth color, but maybe that ambient light environment is constraining. And so figuring out what they do when they can't get that information may be a good, cool direction to go forward. And then I'd say for the frogs, what these really do is they give us a really exceptional opportunity to quantify the costs and benefits of care. And one of the things headed up by my PhD student, Olivia Brooks, is showing some really cool, unexpected stuff. We took different frogs, one that's a facultative egg feeder and one that doesn't have any egg feeding, a congener without egg feeding. We feed them eggs. And we see that the one that gets fed eggs in the wild gains a ton of mass when you feed them eggs. The one that doesn't. Part of this is the one that doesn't get fed eggs doesn't eat all that much. But even when we feed them more detritus food, we don't worry about eggs. The one that gets fed by mom puts on mass much higher rate when you give it more food. So this is our not egg feeder. These are our two different diet treatments. You can probably guess which one got the high treatment. It's this guy over here, but this other species, you can definitely guess who got the food. And these are full siblings, comparable ages, much bigger difference. So we think this is maybe important to explaining the evolution of care as well, is what offspring do with food. So I see I'm running a little late here. So just mention that lots of this is lots of other people. This has been a delight to finally present work for my graduate students where I really didn't do very much. So that's been a real pleasure for me. And of course, Mike and Yusan have been great to work with the whole time. So a little over, let me stop here and take any questions if I can.
All right, thanks Dugas for the wonderful seminar. I was just talking with Mike and you before this that I have actually never heard your seminar, and I was really glad. I'm really glad that I did today because it was pretty awesome. So well, because the YouTube is behind us a couple of seconds, I'm gonna wait for the questions to roll in while I ask my first question.
Okay.
All right. So I'm interested in the kind of curve of the additional benefit that the different stages of offspring is gaining as they grow up. So I'm interested in whether that curve, I guess I guess not that question. I'm gonna like re-ask that question in a different way. So I'm just interested in the kind of like in the parent side of things. Like if parents want to decide to allocate to which offspring, is that strategy expected to change depending on how much resources the parents get to spend? Like if like in a better year or a worse year for the mother bird, does that change their strategies from prioritizing the better offspring as compared to prioritizing some, but also giving enough to allow survival of the other offspring?
So I think you've hit on the place that the begging literature is going right now. And that's thinking about how these strategies are context dependent. And it's really easy to think about in terms of life history evolution that, okay, you have a lot, and then you start spreading your resources out more evenly. And if you're in a food crunch, you want to favor the big offspring. I think the question for me is approximately how this is going to work. Right now, there's a lot of hand waving that, oh, they should switch strategies. And I think that what we're going to want to see is exactly how parents switch strategies. So one of the things I can answer your question, though, is Peter Luther, who's in Chicago, has house sparrow data for like 50 years. And so what having all those data gives him is a bunch of cicada eaters. So he gave me this data back when I really didn't know what I was doing, analyzing it. So I can't give you, so that's why it's not a paper. But what we found is that in those cicada years, you get less mortality, so they don't die. And another side of this is everybody's even sized. So the size variation within the brood goes down when they get during those cicada years. And before you ask, yes, a nestling house sparrow can eat a cicada. I have video of it. It's remarkably easy. It's unbelievably easy for a little house sparrow nestling to eat a cicada. I don't believe it, but I have video of it.
Thanks.
Yeah.
Awesome talk, Matt. So we have a few questions coming in now. So the first one is from Brett Seymour, who's a postdoc with us here at the LAC. And he says, really great talk. Too bad you are not here for full discussion and conversation. And he asks, do you think that cavity nesting birds may have greater low light visual abilities compared to non-nestlings? And also, where's the next part? Shouldn't cavity nesting beggars have more achromatic flanges to increase their signal?
So let me take the second part first. Yes, they do. So that is a thing that we see is there's all these attempts to look across do the comparative efforts. And the one thing that shows up all the time is that cavity nesters have more of this cream yellow mouth parts. They don't have super orange, and those deep reds are missing from the cavity nesters. So that is the one pattern that shows up all the time. One of the things that Shayna is going to work on, Shayna Border, PhD student in my lab, is this idea that we have that maybe it's not so much about being more detectable, because it looks like parents, like even that one where I painted them black and white, like they could see them fine. They still deliver the prey item. It takes a little longer. But maybe what it is that there's just no value to being yellow, because birds aren't actually as good at seeing yellow as we are. And in those lighting conditions, they may just not be able to tell the difference between a really intense yellow and a not so intense yellow. And so maybe that explains it more than the detectability. As for the vision, I don't know if, because the nesting is such a short period of time for them, I don't know if their visual system is likely to be adapted to it, because again, they're going out to catch prey all the time anyway. But I think one of the things I'd like to figure out from a visual perspective is what happens during that transition. Because if you walk in from outside into something equivalently dark to a nest box, I mean, I don't, maybe you, but can't see anything for a little bit, A couple of weeks ago, you're out shoveling snow and you come back in, you can't see anything. So the fact that these birds managed to do that is impressive. And I guess I wouldn't be surprised if there's something there about that transition.
Great, thank you. So we have a next question from Kelm, and the question, there are two questions. So I'll read the first one. We can come back to the other one. So the first one is, could a potential factor for the UV or in general the signal evolution be influenced by brood parasite coevolution? So I'm guessing it's asking the evolution of that flange coloration in brood parasites. Are there any studies on this?
Yeah, so the sparrows don't get parasitized a lot, but there's actually a polymorphism in brown-headed cowbirds where their flanges are really pale and really yellow. And as far as I know, it doesn't seem to matter. So it doesn't seem like there's anything, or I don't know if it's polymorphism, but there are maybe different populations. I can't remember. I think it's different populations of cowbirds are different colors, but it doesn't seem to be about matching them. There's some evidence that cuckoos are sort of a little more elaborate, so a little oranger. They're definitely bigger and shaped differently. There's the really cool one is the Horsfield hawk cuckoo, which is this bird that has wings that look like gapes. So they have little outlines, but that's more about number than matching the color. So there is, of course, there's the Astrobid finches where they have these really crazy things. But whether that's really about their brood parasites, not really clear, because where they don't have brood parasites, they're also really crazy. They have, if you've ever seen the ones with like the blue balls and the like real, a zebra finch has these like white, it does look just like a bullseye. But broadly, it doesn't really seem to. And I think that might just be because all these birds kind of look the same. So there's not that much, right? So it's not, I don't want to say they're all the same, but like aside from the Astrobids, there's not this huge variation, right? They're all kind of yellow. They're a lighter flange outlining a darker mouth. It could be yellow or red or some sort of cream, but it's not that different.
Awesome. So Leanna Van Zen asks, well, she first says, thank you so much for presenting. And then she asks, do you know of any connection between these very visual cues and vocal cues given by the nestlings? I'm also really interested in how this is seen in parasitic nestlings, like brown-headed cowbirds and whatnot.
Yeah, so hopefully I got the cowbird one. But yeah, everybody always asks this, and I've never done it, and I definitely have to do it, I guess, at this point. My resistance to this is that I often think like, okay, you're in the box, you're in a cavity nest, like I don't know if I believe a parent can sort of match a call to a individual bird. I mean, I'm sure they can, but it seems a lot easier to me to match the mouth to the mouth. But no, so I don't know, but it's become very, very apparent over the years that I need to go ahead and figure that out. And so I will let you know when I figure it out. I've been stubborn about it. It's gone on too long. I take that money.
Okay, so we still have some questions in the chat, but unfortunately we are running out of time. So we will wrap up here today, and Dugas, I'll send you those questions later. And if anyone wants to reach out to Dugas for questions, I'm sure he's gonna be really happy to answer via email. So thanks again, everyone, for coming. And thank you, Dugas, for a wonderful seminar, and we will see you all next week.
All right, thanks, everybody.